University of Texas in Austin, and a research associate at the Smithsonian Tropical Research Institute in Balboa, Panama. It would take too long to summarize all of the awards and recognition that Mike has. I needed to get a binder clip to put his CV together when I printed it out today. But just a few of the recent accomplishments. So he received the Joseph Grinnell Medal at the Museum of Vertebrate Zoology at UC Berkeley, the Exemplar Award from the Center of Integrative Studies of Animal Behavior at the University of Indiana. He's an elected fellow and the current president of the Animal Behavior Society, and a fellow of the American Academy of Arts and Sciences, and received a Guggenheim Fellowship in 1997. His book, The Tungara Frog, is now a classic in the field, and his work on the Tungara frog and his relatives has established this as a model system in studies of sexual selection and animal communication. He's published over 219 scholarly articles, and in just the last 5 years alone, he's given 24 invited lectures and 16 distinguished lectures at universities across the world. So we're pleased to have him giving a distinguished lecture here. It's hard to summarize his contributions to the field of animal communication, but one of his major contributions was really shifting the way people think about animal communication away from a view of sort of altruistic conveying of information or sharing of information, and towards more of a view of communication as the signaler trying to influence the behavior of the receiver using whatever means possible, including the exploitation of sensory biases in the receivers. And that was a fundamental shift in the way we view animal communication and how we think that signals might evolve. So that was a major contribution to our understanding of animal communication and sexual selection. His work is truly integrative. His work on Tungara frogs and other model systems has spanned from neuroethology, gene expression patterns in the brain, sensory physiology, up through the ecology of the organism, and looking at broader evolutionary patterns using the comparative method. So without further distraction, I will introduce or let Mike get started. He's going to be talking today about sexual selection and communication in Tungara frogs, brain behavior, and evolution.
Well, thank you very much, Gail, and thank you for the invite. And it's an honor to be invited here to give this lecture, but also it's always an honor to be invited to Davis. I'm not sure how many times I've been here, probably 6 over the years, and I don't think anybody does ecology, evolution, and behavior better than UC Davis. This is an amazing group of people here, but I'm sure you all know that. So what I'm going to do is I'm going to give a general overview of a series of studies that my friends and I have been doing, asking questions about the function and evolution of a sexual communication system. And what I want to do throughout this talk, kind of the subtext, is the subtext of integration. So my main interest is in animal behavior, and specifically in animal communication and sexual selection. And one of the questions that we're interested in behavior is, what's the adaptive significance of behavior? And particularly in these kinds of studies, how does sexual selection promote the evolution of this amazing diversity of phenotypes that we see throughout the animal kingdom? After conducting these studies in animal behavior and behavioral ecology for a number of years ago, it became clear to me that to gain a deep understanding of behavior, we really needed to know something about the evolutionary history of behavior, because the evolutionary history of behavior has a very, very strong influence on biasing the kinds of behaviors that are likely to be evolved, and also in constraining some particular potentials for behavior to evolve. So we've tried to incorporate, a number of years ago, we tried to incorporate very explicit phylogenetic aspect to these behavioral studies. After incorporating the phylogenetic aspect to the studies, it also became clear to us that if we really want to understand behavior, we have to remember that behavior, in some sense, is an emergent property between the interaction of the internal physiology and neurobiology of the animal with the external environment. So in these particular studies of sexual selection, we're very interested in understanding what are the neural and the cognitive mechanisms that are underlying female mate choice, and how do these particular mechanisms, these particular aspects of the brain and of the animal's cognitive system bias the kinds of mating signals that can evolve. Okay, so we make the argument that to get a deep understanding of behavior, we really need to integrate studies of evolutionary history and studies of neural mechanism, and I'm not going to talk about our studies of ontogeny. But this harkens back to Nico Tinbergen's 4 questions in animal behavior, where he pointed out that we can ask questions about survival value, evolutionary history, ontogeny, and mechanisms. He used different words. But what a lot of people forget, or they actually haven't read through Tinbergen's paper, at the end of that paper, Tinbergen points out, but there's really only one question that we're interested in, and that is, why do animals behave the way that they do? And that's what we're interested in here. Why do these sexual selection systems work the way that they do? So frogs end up being a very good model system, I think, for asking questions about acoustic communication and mate choice. And the reason is that it has a relatively simple vocal repertoire. The mating call is under very, very strong natural selection because these calls are species specific and they signal species status to the females. And the females attend to the vocalizations to choose males of the correct species for mating. But the mating call also varies amongst individuals. It's under sexual selection because in most species, females also attend to variation amongst conspecifics and choose some variants over other variants. The mate choice behavior, and I think this is an important character, the mate choice of the behavior is very robust. It can be elicited in the lab, and it's very repeatable. We actually know a lot about the brain, especially about the auditory system in frogs. So it's a very good system to understand the neuroethology of sexual selection and mate choice. And the phylogeny of neurons is more or less resolved. And it seems to be an easier system to get phylogenetic resolution than, for instance, in birds, where the radiation in frogs wasn't as abrupt as it was in birds. So we've been studying this system for quite a while. And again, the questions that we're asking range from phylogeny through behavior and include laryngeal morphology, auditory system, endocrinology, genetic brain imaging. And obviously, I'm not going to have time to talk about all of this. So what I'm going to do is kind of go through several layers of these studies to give you some idea of what we're doing. And I keep saying we. There's a number of people involved in this research. This particular project that I'm going to talk about began in 1986 in a collaboration with Stanley Rand, who was a staff scientist at the Smithsonian Tropical Research Institute. And Stan passed away a number of years ago, but his imprint on this research will be forever there. Walt Wilczynski is the person with whom I collaborate on the neurobiology, and David Canatella is the person who's responsible for most of the phylogenetics and the population genetics. And during this talk, I'm going to highlight some neurobiology work by Kim Hogue and behavioral work by another former postdoc, Ryan Taylor, and then by 2 of my graduate students, and I'll point out their work as I'm talking about it. We have had tons of assistance in this work. Every year we hire anywhere from 2 to 6 people to work with us, and we get this nice mix of North Americans and Latin American students to work with us in Panama. And we are able to collect large amounts of data, and it's because of all these incredible assistants that we have working for us each year. So what I'm going to talk about today, there's going to be 3 parts to the talk. The first part of the talk is going to talk about sexual selection, natural selection, and mate choice in these frogs, and end with a little discussion of sexual selection and speciation. Then I'm going to go over some of the data that we have on how the frog's brain determines what kinds of calls are attractive and not attractive. But then what I'm going to concentrate on are ongoing studies, which we refer to as cognitive mechanisms in mate choice, where we're asking how frogs are making mate choice in complex social environments. So this is the Tungara frog. This is a very small animal. It's only 30 millimeters, its snout to vent length. This is a picture of Barrow, Colorado Island, where I began these studies in 1978 as a graduate student. And these frogs, this is a South American animal. Most of its range is within Central America, but the genus is a South American genus. There's about 40 species in the genus, and the Tungara frog, Phycelamus pustulosus, invaded Middle America from northern and western South America. Now their distribution in the south extends from, there's these frogs in Trinidad. They extend out into the llanos of Venezuela and perhaps up onto the Guiana Shield. Then they're found throughout most of the lowland rainforest of Middle America, and the northernmost distribution of these animals is in Veracruz. And these are all sites where we've worked with these animals. We did a 5,000-kilometer transect through their range to look at population variation. And all of the other close related species are in South America. This is a phylogeny of populations in the closely related species. Everything above node 2 are Tungara frogs. These are their two closest relatives, Physalimys petersi and Physalimys freibergi. And then down at the next node is a number of species, all which are on the western side of the Andes. So pustulosis is South American, Middle American, petersi and freibergi are Amazonian, and then the emergence of the Andes about 15 million years ago divides the western Ecuadorians, the western species from the eastern species. Now what's interesting here is that the species Physalimys pustulosis falls into two very large genetic groups. It looks like the Tungara frogs invaded Middle America about 9 million years ago. And then it appears that there is perhaps a second invasion that perhaps occurred when the Panamanian land bridge formed. So the Tungara frogs started invading Middle America before the formation of the land bridge, and then there's another invasion after the land bridge. So what we end up with, as you can see on this phylogeny, are two genetic groups, and there seems also to be a third group here, but two main genetic groups. One goes from Costa Rica up to Mexico, and the other from western Panama into South America. And one thing that's interesting, this is a structure map based on microsatellites, and this is a map that's based on mitochondrial haplotypes. And in both cases, what we see that there's very strong genetic differentiation between these two groups. So this is Costa Rica on up, and this is western Panama on down. And when we do a haploid map, we see in this same very strong divergence. From what we can tell is that it looks like these populations have had little contact in about 6 million, for about 6 million years. There's a 200-kilometer gap in Costa Rica that keeps these two groups apart from one another. However, when we look at the strength of female mating preferences for the local call versus a foreign call, each of the calls of the 30 populations that we sampled from Mexico on down through South America, there's no relationship between this Ney's genetic distance measure of genetic divergence and the strength of female preference. So what we see is that the species has divided into two different genetic groups. We're calling them the same species. At some point right now, that's a little bit semantic. But when we ask the females if they're the same species, the answer is clearly yes. The females show absolutely no tendency to discriminate against individuals in the other genetic group. Now, this is in stark contrast to a recent study which we did with the sister species, Physalimys petersi. And this study is by Chris Funk, Kat Darst, who had been an undergraduate here, and Kathy Boole. And what we find here is that over a distance of 20 kilometers on either side of the Rio Napo here, between La Selva and Yasuni, and here's Yasuni here, and La Selva would be right about here, we see that these animals have almost 100% unanimous preference for the local population versus the foreign population, but the genetic divergence here is only about 1/3 that it is between the two genetic groups of Physalimys pustulosis. So this is a study of phylogeography and the potential of sexual selection to drive speciation, and it's a situation that we don't understand now. It seems that the pustulosis system should be well set up where sexual selection should have been able to drive divergence, especially given the 6 million year isolation between the groups. We don't see it, and there's been much less genetic divergence here, and we have other data to show that the Rio Napo is not an important geographic barrier, but we see that there's strong divergence there's strong reproductive isolation between these two populations. Now, getting back to the sexual selection within the Tungara frogs, I just want to go over a little bit of their natural history. So, as I said, this is a very small frog. It's common throughout the lowlands of Middle America. It breeds during the rainy season, which in Panama begins about in May, and it continues to November or until December. Males call in choruses, basically in leks. And males don't defend resources, and they call until a female enters the chorus, and the females have full potential to choose mates. So once the female chooses a male, she swims amongst males. She sits in front of one male, moves on to another, moves on to the other. Then she just moves into the male, and the male clasps her. And this is called amplexus. The male's on top, the female's on the bottom. They stay in amplexus for a few hours, and then they make this foam nest. And the foam nest takes about an hour to produce. It actually has, finding out now, it has insecticides in the foam. This isn't our work, but they think it might keep some insect predators off of the foam nest. The eggs stay in the foam nest. They hatch out in about 3 days. The tadpoles develop in 3 weeks. It takes 6 months for them to mature, and the frogs only live 1 year. We don't get recaptures between seasons, and you'll see the reason why. Okay, so what's interesting about these guys is not the details of their reproductive ecology, but the details of their sexual advertisement. So there's about 6,000 species of frogs. Almost all of them vocalize, and all of them that vocalize have a call that is specific to the species. One can walk into the forest, just as one can do with birdsong. You can walk into the rainforest in Panama, and the 30 or 40 or 50 species there you can recognize by ear. You can recognize them much easily by ear than by looking at them. So this is the mating call, the Tungara frog. Now it's a little complex for a frog in that it has 2 different components. The first component is a whine, and this is a waveform showing how call amplitude changes with time. This is a sonogram showing how call frequency changes with time. So whenever a male gives the mating call, he gives a whine, but then to the mating call, he can add these other components, which we call chucks. And they can add up to 7 of these. And this is what it sounds like. And as I said, they can add up to about 7 of these. This just shows the transition probabilities between all the different call states. And the males cannot skip randomly from no chucks to various numbers of chucks. They almost always go from 0 to 1 to 2 to 3, and then back down again. It's not clear why they do that. If they have to do that, perhaps for biomechanical reasons, we're not sure. So when the males call, if a male's calling by himself, he produces only a whine. You walk out to a chorus of males, all of the males are producing some chucks. As far as we can tell, all of the males can produce chucks. Not only in the populations we study in Panama, but when we did a transect through the entire species, we always found that the males could produce chucks. So then the question becomes, well, then why do the frogs make complex calls? By complex, we mean calls with chucks or calls with more chucks. So since this is an advertisement call, and this is old work, the obvious hypothesis is that males are making themselves more attractive to females. So this is where the strength of of Anurin and also cricket mate recognitions, mate preference studies come, in that one has very strong control over synthesizing stimuli, and the animals readily show phonotaxis. They hop towards speakers. And the only reason a female frog would approach a call, certainly in this species, would be to assess the male that's giving the call, to assess that male as a potential mate. So we collect the females in the field in Panama. We catch amplex females. We take the males away from the females. We bring them into an acoustic chamber. Very simply, we shut the males in the acoustic chamber. We observe their behavior through an infrared camera. And then from each speaker, we play calls. So we give females a choice between 2 calls in the standard test, and the females will approach one call instead of another call. So the first experiment we did was we asked, do males gain a sexual selection advantage by making complex calls, calls with chucks, relative to simple calls, calls without chucks? And the answer is yes. There's about an 85% preference for calls with chucks to calls without chucks. And our sample size, this is an old slide, our sample size now is about 5,000, and we see that every year we get about the same strength of preference. It varies between about 82 and 86%. So it's a very, very repeatable preference for complex calls. So if the males increase their ability to attract females by making complex calls, then why don't they always make complex calls? And this is the answer. It's a very dangerous thing to do. This is the frog-eating bat, Trichopterosis, and it's unusual in a few ways. One is that it eats frogs, which is unusual for a bat, not unheard of, but unusual. But the way it catches the frogs is by localizing the calls of males. So when males are calling to females, they make themselves more attractive to these frog-eating bats. And these bats do have an echolocation system, but they don't acoustically image their prey using the echolocation system. They find the frogs mostly with passive localization, and they have a series of adaptations in the inner ear that allow them to extend their hearing range from the ultrasonic frequencies down to the frequencies that frogs produce. So in that sense, as far as we know, this bat is unique, having those auditory adaptations. If you play to the bat the simple whine call, like the female frogs, the bats approach the whine. But if you give them a choice between a simple call and a complex call, the bats prefer the complex call. These bats are very common in Panama, and they eat a lot of frogs, but that's not the only problem that the females have. There's also a fly. This looks like a mosquito, but it's not. It's a fly in the group Corythrella. This is the sister taxon to mosquitoes. And these flies take blood meals from calling males. My graduate student, Jimena Bernal, has shown that the flies are attracted to the male's calls. And when you give the flies a choice between a simple call and a complex call, the flies also prefer the complex call. Now, what's interesting here is we don't know, these flies should not be able to hear these frog calls. But when we first discovered trichops, we were told that this wasn't possible because bats couldn't hear low-frequency calls. So Ron Hoy, who's an auditory insect neurobiologist at Cornell, Ron has looked at these flies, and he thinks that these flies have evolved a unique ear that allows them to hear frog calls. Now, there's also flies that orient to cricket calls, and the females oviposit on the crickets. Those flies, Ormia, have evolved a unique ear, and Ron Hoy was the person who discovered that ear. This, he says, is a different ear, appears to be another innovation that allows flies to acoustically locate prey. And as I said, the flies do the same thing. They prefer complex calls to simple calls. So what we see then is very simple example of how sexual selection and natural selection generate conflicting forces on the advertisement call. And these were the early studies that got us interested and committed into using and exploiting this system for studies of communication, because we were very happy about how we could look at not just the mate choice advantages of communication, but also the natural selection costs of communication. And again, one of the early important results for us was this fact that the females prefer complex calls to simple calls. So we decided that that we can demonstrate behavioral preference for complex calls, what we would like to do eventually is to understand the evolution of these preferences. We've mapped out these behavioral preferences on the phylogeny, and it appears that the preference for complex calls actually evolved prior to the complex calls themselves. But I'm not going to talk about that research now. Instead, what I want to talk about are some of the studies that we've done of the brain, trying to understand where in the brain does this preference for complex calls reside. And a recent postdoc, Kim Hoke, who's now at Colorado State, Kim, in collaboration with myself and Walt Wilczynski, conducted a series of studies looking at the response of the brain to variation in their exposure to advertisement calls. And while we use gene expressions, we've done electrophysiology studies, but to look at the peripheral auditory system, but to look at the brain, it appeared that using gene expression studies was a much more powerful approach. And I know that a number of people in the audience are quite familiar with how these immediate early genes work. So I'll just briefly summarize. There's a class of genes, immediate early genes, whose exact function appears not to be known, but these genes express themselves when there's action potentials in a synapse. So they express themselves in response to excitatory, although not inhibitory, action potentials. So what we do, meaning what Kim has done, is we expose the brain, well, in the female, we expose the female to call variation. And what we want to understand is how does call variation influence mate choice? Now, there's different functional steps to mate choice. The animal has to receive the signal. There's auditory processing of that signal. That then leads forward to a decision, and that decision then has to influence motor output in the animal. Now, you'll have to take my word for this, because I don't have time to go over these studies, but what we have shown is that the auditory processing is taking place in the brainstem, and in the brainstem, and these are the data I'll review, there's enough information there for females to discriminate between conspecific and heterospecific, and simple conspecific and complex conspecific. We show that there's a social neural network in the hypothalamus, and coordinated activity of neurons within the hypothalamus are elevated when the animal receives more biologically salient and attractive signals. And then that information is fed forward to the telencephalon, where these motor patterns that result in phonotaxis are activated. But just to give you a flavor for how these studies are done, a female is exposed to one of 4 different signals, either the simple whine, the complex whine. This call is of another species of Phycelamus NSFE, that's ironically named for the National Science Foundation, or they can be exposed to no sound at all. So after the females hear the song for half an hour, these are done in Panama, the females are firstly taken from the field, they're sacrificed, the brains are frozen, we bring them back to Austin, and then whenever I say we, of course, I don't mean me, I mean them. And then Kim does in situ hybridization. She slices the brain, does in situ hybridization. Then you can identify where there's messenger RNA for the immediate early gene. And this gives you some indication of the amount of neural activity in different parts of the brain. Now, in the frog's brain, right here, right below the optic tectum, is a very large auditory nucleus called the torus semicircularis. And for the neurobiologists, this is homologous to the mammalian inferior colliculus, where we start to do most of our auditory processing. So the first study that we did was to look at the amount of gene expression in the hindbrain, in the torus semicircularis. And what we found right away is that the amount of gene expression, and therefore it should be neural activity, changes drastically and substantially with the calls to which the females are exposed. So you get more activity in the auditory nucleus when they're exposed to complex calls, less when they're exposed to simple calls, and even less when they're exposed to heterospecific calls. And I want to point out that all of the energy in this heterospecific call is within it overlaps the frequency range of the Tungara frog. So the females are getting auditory input, and this call has a lot more energy. So this isn't merely a difference of a complex call having some more energy than a simple call. And then you have the least activity when they're silent. So what we see then is this is where in the brain that females discriminate between conspecific and heterospecific calls, and between attractive calls and unattractive calls. At the auditory nuclei below this, the superior olive, we don't get any difference in gene expression in response to these signals. So that's what the brain tells us. And as I said, what we've been able to do is map out the series of connections throughout the entire brain to give us at least a hypothesis of all the steps in the mate choice decision-making process. It's very descriptive, and it's a very difficult hypothesis to test experimentally with these kinds of animals. It's a very correlative description of how we think information is processed in the brain. But it matches very well recent studies on social neural networks in birds and in mammals that have been done quite independently from this kind of research. But what I want to do now is I want to really switch gears for this last part of the talk, and I want to acknowledge that the experiments that we've been doing, including the gene expression neuro studies, have been asking some very, very simple questions. But we need to remember that these animals are operating in socially and in environmentally complex communication systems. So here's a koala frog. So through the neuro studies, what we've really been doing is taking a bottom-up approach. The idea is that if we can understand the response properties of the neurons to sound variation throughout the entire brain, that's going to lead us to understand how mate choice occurs. Now what we want to do, though, we want to ask some different questions about how animals can operate in a very complex communication scene and extract meaningful information from that complex auditory and, as you'll see, visual scene, and how that influences their mate choice. And so we're there, now we're going to take a very top-to-bottom approach. And what our hope is, by continuing the neuro studies, working our way from the bottom up, and then beginning these studies of what we call cognitive mechanisms in mate choice, working from the top down, that at some point these two approaches are going to intersect, and there will be an epiphany, and then we're going to stop working because we'll understand everything. So when we think of, we call this series of studies trying to understand cognitive mechanisms of mate choice, and this is using cognition in the sense of the need to acquire energy, acquire information and analyze that information. So we're not at all suggesting that these frogs have a theory of mind, that they're introspective, that they're thinking about what they're doing, but we do suggest very strongly that they're analyzing information. And even our more reductionist studies of the brain do show that in reality they are analyzing information, and we can even touch the part of the brain and show where they're doing it. So what motivated us to take this approach and start looking at these more cognitive mechanisms were three things about the mating system, about the communication system, that we realized we weren't addressing in other calls. One is that calls are ephemeral. They're not like some visual signals that are turned on all the time and females can continually assess them. But calls are ephemeral. Calls change during a call bout. So the call bouts are dynamic. So the females don't have access to the same information from call to call to call. And then the last point that we'll end up on are calls, but they're not just sounds. There's other modalities involved in calling. So this has led us to ask questions then about, in the first case, about active time and memory. In the second case, about Bayesian or Bayesian-like updating and decision-making in mate choice. And then we're going to end up talking about multimodal communication. So as I said, calls are ephemeral. Frogs and insects and birds often chorus in what are called unison bout chorusing. They call for a while, and then they stop. And then they call for a while, and then they stop. And these are just some examples of chorus bouts of the Tungara frogs in Panama. And the average duration of the choruses are somewhere between 30 and 40 seconds. As you see, most of them are most of the bouts are less than 30 seconds. Here are between 30 and 60 seconds long. And the average, I think, is about 45 seconds. And then these are separated by bouts of silence. And the interchorus interval is usually anywhere from a few seconds to up to 30 seconds. So what we wanted to know is, when the males stop calling, do the females have any memory for what they've heard? Or do they need to begin their assessment of call variation all over again? Another motivation for asking that question is, we have shown that there's very strong preference for calls with chucks to without chucks. So for example, if you give a female 0 chucks versus 1 chuck, in this study, 15 females went to the call with 1 chuck, no females went to the call with 0 chucks, 20 females go to the call with 2 chucks, a few females with 1 chuck. But if you give them more chucks versus fewer chucks, there's not significant preference for calls with more chucks versus fewer chucks. So we thought possibly the function of more chucks is that it instantiates longer short-term memory in the females, and therefore promotes the active time of the signal. Now, if you go to a communication book or if you Google active time, there's no definition of active time because we've just suggested this idea, and it comes from the idea of active space. Any signal has an active space defined as the space or the volume over which that signal influences the receiver. And we know that there's various factors that can influence the active space. If the animal's calling in dense forest, then the active space will be less because of attenuation of the sound through the environment. If it's a vervet monkey calling on the top of a canopy during a thermal inversion, then there's actually negative excess attenuation. The calls of the monkeys are beamed farther than they would normally travel based on what we would expect from the physics of the signals. So we hypothesize that the same thing might be going on with active time, that we assume that for any signal, the probability of remembering from where that signal emanated is going to decrease with time, and we assume that there's things about the signal that can enhance the memory decay of the receiver, make the signals less memorable, or that can enhance memory, that is, make the receivers remember the signal for longer. And this idea that signals can enhance memory of receivers is not our idea whatsoever. At least, I mean, this goes back actually to some early studies that Richard Dawkins did when he was a faculty member at Berkeley. But a little bit more recently, Marion Dawkins and Tim Guilford have talked very broadly about receiver psychology and trying to understand the evolution of warning coloration. And they point out that some colors and some combination of colors are remembered by predators longer than others. So this idea that we have about active time and signaling is really derivative of these discussions that Dawkins and Guilford have been having for a number of years. So specifically what we wanted to know is do females have more memory for longer memory for more complex calls? And this is a very simple experiment. Females are in the middle of this chamber. They're under a funnel, an acoustically transparent funnel. One speaker plays a whine. The other speaker plays a whine chuck. The funnel is raised. I'm sorry, the female's under the funnel. And then there's a silent delay period. And then the funnel is raised as calls are played, but now we play the identical call. So there was a simple call here, a complex call here. The calls are shut off. Then the female is allowed to choose between two simple calls. If there's no memory, the null hypothesis is that she should choose randomly between these. If there is memory, the female should prefer this call versus this call. And these are identical calls. We do this for calls with 1 chuck, we do it for calls with 3 chucks, and we vary the delay time from 0 seconds to 30 seconds. What we see here, if we look at the results for the 1 chuck call, this is the black bars are the speaker that had been broadcasting the complex call, but is now broadcasting the simple call when she chooses. There's no effective memory whatsoever. If we look at the 3 chuck call, there's an effective memory at 0, 15, and 30 second silent delay. So the more complex call, but not the less complex call, has a greater active time, and that's because it enhances the memory of the females. There's other alternative hypotheses, but we've tested those alternative hypotheses, and we've been able to reject them. So we're left with the conclusion that the effect is actually on memory. Now, if we titrate the active time of the effect of the 3 chuck call, what we see is that, again, there's a memory effect not only out to 30 second delay, but 45 second delay. There's still a trend for a memory effect at 60 seconds, and by 2 minutes, there's no effect whatsoever. So what that shows, though, is that females have memory out past 45 seconds, getting towards 60 seconds, and that would encompass a very large percentage of the interchorus intervals. So the female's memory for signals is enough to allow her to remember more complex calls from one chorus to another chorus in almost all cases. So what we see now is that we have shown that females prefer complex calls to simple calls, Even though there's no consistent preference for more chucks to fewer chucks, we think that there is actually an advantage to more chucks, and that's because it instantiates more memory and makes the calls more memorable to the females. And we will make the argument that this might be a general explanation for the evolution of signal complexity. Certainly not in all systems. There's a lot of reasons why animals can evolve more complex signals, but this idea of memorability has not been suggested before, and we suggest that this actually might explain why lots of animals evolve to have more complex calls. Okay, so calls are ephemeral over long periods of times, but call attractiveness also varies within the call bouts. So a male can call this male, for instance, he can call whine, 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 where some males will escalate to 1 chuck, then go back to a whine. Other males will increase and then plateau at 2 chucks. Other males will go right up to 3 chucks very quickly. So females are listening to a call. They prefer complex to simple calls. These calls are changing dynamically. When do females make decisions? Do they update their decision and change their mind? And are there any internal physiological features that might influence when they change their mind, how they update? So there's a very famous paper in 1979, and this is the title, "Don't the Girls Get Prettier at Closing Time? A Country Western Application to Psychology." And what Pennebaker pointed out in this study is that if you go to a bar early in the evening and you ask members of one sex to assess the attractiveness of members of the opposite sex, and if you do it early in the evening, then if you do it right when the bar is going to close, people's assessment of the exact same individual of the opposite sex increase as the bar gets closer to closing time. What we have shown in a study I'm not going to talk about, a study by Kathleen Lynch, is that as the females get closer to dropping their eggs, which we have shown is correlated with higher estrogen levels, and we can mimic this effect by injecting females with gonadotropins, that when females are closer to dropping their eggs, they will show phonotaxis to signals that they would never show phonotaxis to before. So this is what we do, this very simple and fun experiment with the frogs. The female's put in the middle of the arena. She's given a whine and chuck from one side, a whine from the other side. We call this our decision boundary. The females approach, and now when they get to this decision boundary, what we do is we take the calls and we now flip them between the speakers. So she was headed to a whine chuck, and now all of a sudden it's a whine, and the whine chuck is coming from behind her. And when we flip the calls, we do 2 different kinds of experiments. In one experiment, we just flip the calls and we leave them at the same amplitude. So now the whine to the female will be louder than this one, just because it's closer. Or in another experiment, we boost the amplitude of this call, so at the female here, it's the same amplitude. And then we simply ask, what do the females do? Do they update their decisions? Do they change their mind? Or do they continue committed to the same male that they were approaching? And the answer is that they do both. Sometimes the females stay committed, and then sometimes they show what we call reversal. And we've done a very large number of experiments. I'm only going to talk about a few of them. And what we show here basically is that if we do a control, the initial stimulus is there's a whine chuck, and then in the control we don't switch them, but we boost the amplitude of the less attractive call, which is behind the female. We don't see any reversals. If we flip the calls and we equilibrate the amplitude, so we boost the amplitude of the attractive call, which just ran behind the female, so now she hears them, the same amplitude, then 44% of the females reverse. If we don't boost the amplitude, we still get reversals, and there's 30% of them are reversed. So what we see here is that the females do updating. It looks very similar to Bayesian updating, and that the reversals are contingent, in this case, on 2 factors. One is the complexity of the signal, and one is the amplitude of the signal. Now what's interesting is, and we probably shouldn't have done this, but we can ask a female just for fun, like, how many times can we get you to reverse? So if you remember The Exorcist, when Linda Blair's in the bed and her head spins around, so it's kind of like that experiment. So we can just keep doing this, and we had some females reverse 16 times, but then we had other females who didn't reverse. So we did an experiment to look at variation amongst females, and we tested each female 6 times. So she could reverse anywhere, a single female could reverse anywhere between 0 times and 6 times. And if we look at the distribution, we see what looks like a bimodal distribution. So we see, it looks like we have reversible females and we have non-reversible females. Where is this coming from? Well, so we classify these females being less choosy or more choosy. And if we look at the females, and if we plot the female's body mass versus her snout to vent length, what we see is that the non-reversible females, the females who stay committed to that early choice, the non-reversible females are very heavy for their body size here, while the reversible females are light for their body size. They're kind of skinny. And we think what's happening is as females are getting closer to when they're going to drop their eggs, the eggs hydrate and the females become heavier. And therefore, what we think we're seeing here is state-dependent decision-making, where once again, but on a very different time scale, the female's reproductive condition is influencing her decision-making. It's not making her more permissive, as we've shown in other studies, but it makes her less likely to update her decision. Now, in just the last couple of minutes that I have, I want to just tell you about this last set of studies that we've done on multimodal communication. Frogs have vocal sacs. Greg Pauly did some work with us on Tungara frogs and showed that one of the advantages of a vocal sac is it takes the air and it quickly shuttles it back to the lungs so the frogs can call more quickly. If the frogs couldn't do this, the frogs would have to buccal pump the air into their lungs. They're positive pressure breathers, not like us. Frogs don't have a diaphragm, so they can create a greater space that allows the air to rush in. But when frogs call, you can see their vocal sac vibrating. And for people who have been out in the field trying to find frogs, you know that you can get so close to a frog sometime, you can feel the air, their call hitting you in the face, but you just can't see them. And what gives them away a lot is you see the vocal sac flittering. So we wanted to look at the role of the vocal sac in communication. So this is work by a recent postdoc, Ryan Taylor. So we worked with a grad student at Texas, Barrett Klein, and an engineer in New York City, Joey Stein. He's from Brooklyn. That's why his name's Joey Stein. And I'm from the Bronx, so I can say that. So this system, there's these little plasticine models of the frog. There's a pump that's driven by a computer. So when the computer broadcasts the call, it drives this pneumatic pump that inflates the vocal sac. The vocal sac now is a catheter. In the beginning, the vocal sac was a condom, and they would break all the time. So we replaced it with a catheter, which breaks, but not as often as the other material. So this is just showing a real Tungara frog from different angles, and this is the model. And this is the model. So we've asked some very simple questions. The first thing we did is we measured females' threshold sensitivity to light, and specifically to distinguish between the hue of the vocal sac and white using optomotor system. And what we show is that these frogs can see incredibly well in the dark. Not better than other frogs necessarily, but way better than we can. We measured the illuminance of light at breeding sites in Panama, and the females can see fine even when it's a dark, cloudy night. So the females can see this stuff. The way we do these experiments is like all of our experiments, very simple. A female's in the middle of an arena here. There's a speaker here, and the frog, the robot, is placed in front of one speaker and not in front of the other speaker. The female hears the call. We use a light that mimics moonlight, both in intensity and also in spectra. And the female's released, and I'll show you what she does. Oh, we can't hear it. Oh. Holy moly. Can we do this in the light instead of the dark? I'm okay. So can someone do that for me?
The light switches over just by the actor.
Ah. Okay. So I can tell you the results because they're very simple. First thing is that females find this call that's associated with the robot more attractive than the call by itself, number one. Second result is that you can decouple, we can experimentally decouple the call, the acoustic signal and the visual signal. And I don't know if you know in humans there's something called a McGurk effect. So if the lips are not giving you the same, if you have the lips saying ba, but the sound coming out of a video is pa, you don't hear either of those signals. It's an amazing, you can look it up on YouTube. It's a beautiful demonstration. And that was demonstrated in the 1970s by McGurk. So we wanted to know if frogs had something like a McGurk effect, and we found something quite different. So if you give the females a choice between the call and a robot, and again, the robot is in sync with the call, then the females go to the robot plus the call. If you have them out of sync, not 180 degrees out of sync, but just displaced, the call is about 300 milliseconds long. If you have them displaced by about 150, or if they're non-overlapping, you get the same result. If they're displaced from one another, we would think there's two possibilities. It doesn't matter to the female. They're still going to go to the robot and the acoustic signal. Or it's going to disrupt the salience of the signal, and now the females will choose randomly. What it does is it reverses the valence of the signal. So not only do the females no longer prefer the robot with the call, they avoid the robot with the call. So now something's happening where this inflating vocal sac, being a little bit sloppy in terminology, takes on a different meaning to the female. It goes from a positive valence to a negative valence. So that's the last, so that we don't need it anymore. So that's and the last empirical part of the data, the last study that I want to talk about. And then since there's a little bit late, I just wanted to sum up saying that what we've learned about the system is that we haven't learned enough about the system. And when we first started these studies concentrating just on the behavior and mate choice, we thought that we had made really nice strides to understanding how sexual selection and communication functions and evolves. And then by incorporating the phylogenetic approaches and by incorporating the neurobiology, we thought we made more strides. But now, especially with the neurobiology, as valuable as that is, and we're continuing to collaborate on these studies now, but we realize that these frogs are doing stuff that's so much more complicated than we thought they were doing, and we just try to make ourselves go out every year, us and our citizens, we go out and we sit in these ponds for an hour or 2 hours, just to remind ourselves that this is behavior that's not taking place in the laboratory, we're testing it in the laboratory, but it's taking place in nature, and we just have to put our heads and our shoes and our butts in these ponds and watch the animals every year to remind us, just because we can do nice simple experiments doesn't mean we're really explaining what's happening in nature. So these cognitive experiments are trying to move us a little bit closer to that. So thank you very much, and I don't know if there's any time for Oh, okay.
Yes.
Yes?
So is the complexity of the call heritable? Like, have you looked at that? Like, how heritable it is?
Well, we haven't done heritability studies, but what our guess is that the call, it's inherited, but it's not heritable. So all males that we know of can make complex calls. We know how they do it. Their vocal fold has this big mass that hangs from it. And what happens is, and we're working with some engineers now to get a better understanding, this mass, under certain pressure, sets into a different phase of vibration, pulls the rest of the vocal cords with it. Now, the sister species, Phycelamus petersi, some populations make complex calls and some don't. And if you look at the laryngeal morphology, it's just what we predict. You have a big old fibrous mass like a testicle hanging from your vocal cords. If you make complex calls, if you don't, that thing's really small. What we don't understand is the degree to which that's heritable in that species where we actually get variation. But in the Tungara frogs, every larynx we've looked at looks more or less, of the males, looks more or less the same. Females don't have this fibrous mass. So there might be variation, heritable or not, in the propensity of the male to make complex calls. We don't know about that. And that's one of the many, many things that we have to and haven't looked at yet. Michael?
Any insight into that comparison between Peter's eye and tuberculosis as to whether or not the calls have diverged? We did close a wonderful bit of natural history, and I didn't get the answer.
Yeah, so the answer that someone told me, so why did they speciate, why did these guys in the Amazon speciate and these guys in Middle America don't speciate? And they said, because it's in the Amazon, everything speciates in the Amazon. But the real answer is, I mean, we know what happens. So these two populations of Peter's eye, one produces complex calls and the other doesn't. Now, the females show preference for the local call without involving the complex call at all. If you give each of them their standard whine, you get this strong preference for the local call. What happens is when you evolve this big fibrous mass, even when the fibrous mass isn't set into vibration to produce a chuck, it's still weighting, adding mass to the vocal fold. So it brings down the entire frequency sweep of the whine. So we haven't, and this is a testable hypothesis which we haven't tested yet, but what we're pretty sure is that the whines differ in frequency as an incidental effect of one population evolving the morphology to make complex calls, but it also influences the simple call. We know in the Tungara frogs, females are very sensitive to the frequencies and the whine as well as the chuck. So that's our guess. Now how come that would happen? And this has happened multiple times from what we can tell in Peter's eye. It looks like there's 4 to 6, depending on the coalescence analysis we did, how you interpret it. 4 to 6 independent evolutions of complex calls going from Ecuador down through Peru into Bolivia in this species. Now, we haven't shown the female preferences for local calls. We've only shown that in this one case. We haven't studied the others. But we don't know why this behavior and this morphology is so more variable in that species than in the Tungara frogs. And we certainly don't know why the Tungara frogs haven't diverged with this big 200-kilometer gap in a Ney's genetic distance of 0.1, which is where you get most frog taxa species being called species when there's about 10% sequence divergence. So we have a scenario for why it happened in Peter's eye, in the Amazonian species. But we don't know, I mean, we know how it happened. We don't really know why it happened there and why it doesn't happen in the Tungara frogs.
Do you think there was ever a predator out there that tried to attract females by making the male chuck calls, which is why they're so suspicious of disconnects?
Do we ever think so? Yeah. Do we have any evidence? No. What we thought was we found some night monkeys eating frogs. Yeah. So we had this great idea, and night monkeys will eat bats. So we had this great idea that the night monkeys were catching the frogs. This is when I was a grad student. That the night monkeys were catching the frogs and getting the frogs to call up in the canopy, and that the bats were coming by and the night monkeys were grabbing the bats. But when we got sober, we rejected that hypothesis. So we don't know. So that is a really good question. We don't have any examples of that at all. And we also don't have any examples in any neuron of acoustic mimicry. So for instance, I didn't talk about this. These bats won't go to the calls of poisonous frogs. And the bats are very labile in their ability to associate prey palatability with sound. My student Rachel Page, who's a staff scientist now, she's shown that you can condition these bats to go to reggae music if there's a frog associated with that. So these bats are behaviorally very flexible. But we thought that and they won't go to calls of toads, we thought we had found some species that when there was bright moonlight to us, sound like they're changing their calls to sound a little bit more like toads and make themselves less attractive. And you don't see that either. So we don't have anybody imitating the frogs so they can eat the bats, and we don't have anybody imitating distasteful frogs. The only example of neurons where it gets a little bit tricky like that is the frogs have distress. A lot of frogs have distress calls. They open their mouths wide and scream. So if a snake takes not a Tungara frog, snake takes a frog, frog screams. In Costa Rica, caimans come up and take the snakes. On some islands in Lake Michigan, raccoons come up and take the snakes, and the frogs get free.
So my understanding is there are some birds out there where complexity correlates with the quality of the male. In your guys, what correlates with the quality of the male if it isn't complexity?
Yeah. I would actually argue that in most of the studies for birds, there's not good evidence. There's some evidence, but not good evidence. It's very correlative evidence, and there's not a lot of studies that show heritability for, well, forget heritability for true fitness, heritability for physical vigor. And sometimes the arguments get confused when people confound total fitness and include the component of mate attraction. But there's some studies of tits that suggest that might be happening, and peacocks maybe, based on tails. What does it correlate here? I don't know. But I don't know. Ahmad Zahavi was with me when I was a grad student, and Zahavi suggested that the animals, when they were afraid or nervous, that their vocal cords would tighten up and they wouldn't be able to make a chuck. I mean, I tried to think of ways to test that, but wasn't able to. But something that we have found interesting was a student of mine, Karen Akre, who did these memory experiments. I forgot to acknowledge her. What she finds is that if a male is making a call, a simple call by itself, females will come, and what the female will do, they won't swim up to the male and allow the male to amplex them. They swim by the male very quickly, and they stop and they turn around. When the female does that, the male often adds a chuck. Not only that, what she shows is that the female, if the male adds one chuck and not more than one chuck, the females will come over and they'll bang into the male and back off. And they won't choose the male, allow themselves to be amplexed. I mean, this is sloppy language, but they won't approach the male and stay still so they can be mated until they get the male making more chucks. So the females are inciting increased male complexity. So they're, in a way, inciting the males to make more costly calls. And we know they're more costly because not only do the bats prefer calls with chucks to no chucks, I didn't show the data, but we now know the bats prefer more chucks to fewer chucks. And when Karen was doing these studies in Panama, she called me on the phone, and she said, I'm really concerned because, I was doing those studies, and the guy goes from 0 chuck to 1 chuck, and the female hits him. She adds another chuck, the female hits him. And she said, out of the sky, the bat came. She has this on video. The bat comes down and catches the male as soon as he adds 3 and eats the male as soon as he adds 3 chucks. And she was concerned it wasn't in our Ayakuk protocol, which is a really sad commentary, I think, on Ayakuk. You can watch a predation event in nature and somehow think it's your fault. But it does seem, but that is very suggestive of, of like Zahavi's handicap principle that, again, well, I won't say being anthropomorphic, because that hypothesis is framed in an anthropomorphic way, that the males are advertising to the females their ability to survive. And if one were to take Zahavi and extend it, this is what you would expect, right? That the females wouldn't only assess the signal variation that's there, they would be under selection to be able to assess the limit to which the males are capable, able, willing to add those kinds of signals. So obviously we don't know. There's lots of speculation.
How might more chucks be indicative of the ability to survive? Because it doesn't seem like the males have any particular avoidance behavior associated.
Well, no, but they do. I don't think. Zahavi thinks it's indicative. He said it's indicative of their having good genes, and I said it's indicative of them having good luck. And how do you have to discriminate those two hypotheses? But no, the bats the frogs, they can't hear ultrasonics. We know that. from electrophysiology and from behavior. On a dark night, the bats can come right in and pull the frogs right off of the water. And this is a very common event on Barra, Colorado and Panama. It's very easy to see this with a night vision scope. If the sky is clear and the moon's out, and it doesn't need to be a full moon, then the frogs can detect the silhouette of the bats coming. And then they stop calling, and depending on the height, we do this study with models of bats. Depending on the height of the bat, you get more extreme avoidance behavior from the frogs. Then what the frogs do is the bats will shut down the chorus because frogs attend to their neighbors. When their neighbors stop calling, this spreads like a sound of silence through kind of the mega chorus. Everybody stops calling. And then what they do is then they listen. So if they don't hear any other frogs call, they'll start calling in about a minute or 2 minutes. If you play them a Tungara frog call, they start calling right away. If you play them a call of a local heterospecific frog, it gets them to start calling again. You play them a call of another Physalema species that's from South America, has no effect on whether they start calling again or not. So they seem to be listening for what's referred to as public information about predation risk. But I don't think producing more chucks is indicative of their ability to survive, but it could be. We just don't have any data one way or the other.